let's start. So for my bachelor thesis, I made a multi-order adaptive network model for pathways of DNA methylation and its effects in individuals developing post-traumatic stress disorder. So let's start with what is post-traumatic stress disorder. So post-traumatic stress disorder is characterized as having at least one re-experiencing symptom, for example, a flashback, at least one avoidance symptom, for example, avoiding going inside of a car, or because you were in a car crash, at least two arousal or reactivity symptoms. So having a big reaction, big emotions, at least two cognition and mood symptoms for duration of at least 1 month. So mood disorders and such. So what causes PTSD, post-traumatic stress disorder? Post-traumatic stress disorder can be triggered by having some kind of traumatic event or a long exposure to chronic stress. So one traumatic event would be, for example, a car crash, and long exposure to chronic stress would be maybe living in a war zone or living in a space in like a bad neighborhood, for example. What are the therapeutic options for PTSD? Is psychotherapy or exposure therapy is usually considered the golden standard. It is often combined with antidepressants, and basically it works well, but there are a lot of dropouts. There are a lot of dropouts from the therapy, and it's not always successful. So for my thesis, I am considering epigenetic effects that are happening during development of PTSD. And I have chosen to work on DNA methylation as it has been most research of all the epigenetic factors. There are others, such as histone methylation. So what is DNA methylation? DNA methylation is a transfer of a methyl group into the C5 position of the cystine form of 5-methyl cystone. So basically, this is something that is already happening inside our bodies. Our genes already have levels of methylations on it. So what does DNA methylation do? It controls the expression of the gene. So more of so basically it suppresses the gene expression. So more methylation, less expression of the gene. So DNA methylation dysregulation has been observed in PTSD on two different biological processes, on the immune system and the stress response. My model focuses on DNA methylation deregulation on the stress response system and its effects on emotional regulation during a flashback episode. So the model follows an individual with increased glucocortic receptor sensitivity to glucocortics, which results in a low cortisol levels. Okay. So basically, two genes that I have chosen to emulate are NR3C1, and that's the gene that encodes a glucocortic receptor, and FKBP5. And FKBP5 encodes, so it codes FKBP51 protein, and that is the protein that regulates glucocortic receptor sensitivity by creating an intercellular ultra-short negative feedback loop. So basically, it attaches itself to the glucocortic receptor So a cortisol cannot be attached. So basically it suppresses the sensitivity of the receptor. So they both basically, I told you already, but they have a role in stress response regulation. And yeah, so they are most researched in DNA methylation. So for methods, modeling approach is based on temporal, is a networking modeling approach that is based on temporal causal networks. So basically approaches based on your findings in literature. You make a conceptual model, and then you fill out the role matrices. So connectivity, so how each state of the model is connected. And there is a value of that connection. So we have aggregation. So basically each state has its own combinational function that dictates its behavior and how other states affect it. And timing, each state has a speed factor, or that depends how fast the state is changing. So these role matrix, so not role matrices, these, so for example, combinational function, the speed factors and connections are put inside of a function that basically, and the model is run. So for the states, I have chosen 4 different combinational functions. I have chose a logistic function because it has, so basically already a lot of, I have been using, I have been referencing in my paper to different models that have been already made, and a lot of states already there were already used for as a we're using a logistic combinational advanced logistic sum function. And another thing that's good about advanced logistic sum is that it doesn't go above 1 or goes below 0. And it was very good for the gene expression, as for example, glucocortic receptor threshold. I could use the threshold as a self-modeled state that would to show expression and its effects of FKBP gene. So Hebbian learning has already been inside of the model that emulates the mental, that is emotional regulation and the flashback. I have used StepMod, that is a function that repeats itself. So a state constantly either is 0 or it's 1, and it repeats itself every 100 timestamps. And step 1's function is used for the traumatic event occurrence, and it is used for and for therapy, because they both happen at one prolonged period at one moment. So adaptiveness of the model is achieved by making some of these network characteristics another state, so-called self-model state, that represent that characteristic. Therefore, so basically a characteristic is now, it can change basically. So this is called a first order adaptation, and if a state in first order adaptation uses an adaptive characteristic also, the state that represents that characteristic would be called second order state, and for the second order it would be third order state, and so on. So this is a conceptual model of my model. So this part, so basically you could divide it into two different models. So this would be a biological side, and this would be a mental side. How does it work? So first of all, everything starts in the mental model where the traumatic event has happened. The traumatic event is censored, and a sensory of that traumatic event causes chronic stress. Chronic stress then affects body stress, and our body is under a lot of stress. Hypothalamus registers that body stress and sends out a protein called CRH to the pituitary gland, and the pituitary gland sends out ACTH to adrenal gland, and adrenal gland starts to produce glucocortics. Glucocortics glucocortics reduce the immune system, and the immune system. So basically, for the immune system is triggered by the infection. Infection is constant, as human body is constantly under effect of some kind of viruses or fungi or bacteria that is constantly attacking us, or some kind of injury. And this injury triggers the immune system, and this is basically how the whole network works. So let's go back to the glucocortics. Glucocortics reduce the immune system, and in return, immune system, when it's triggered, it causes inflammation, and by reducing the immune system, the inflammation is also lower. Glucocortics also affect glucocortic receptors. Glucocortic receptors sense glucocortics, and they tell hypothalamus and the pituitary gland to stop the production of CRH and ACTH, so there are not too many glucocortics, and this is kind of a loop that is constantly happening. So let's continue to the methylation. So methylation is achieved by making five orders of states. So approach I've taken here is that so basically glucocortics demethylate genes, and glucocortics also trigger transcription. DNA repair system is triggered when there is demethylation, and it fixes the demethylation by methylating the genes again. So let's continue to the mental model. The mental model is taken from made by from the paper Disturbed. Actually, it's a chapter in a book, Disturbed by Flashbacks, and it is created by Leila van Mens and Jan Treur. The person senses the traumatic event, and that is here. And basically, sorry, 1 second. So traumatic event happens, and the person, this would be this would be these three states would basically represent traumatic this would be sensory of the traumatic event, and this would be process in the brain of the sensory of the traumatic event. And the person senses the traumatic event, and because it sensed a traumatic event, basically the traumatic event stays in their head as a form of video that continues to so that is basically the memory. So these states affect, well, okay. So these states would represent a trigger that would trigger the flashback. And for here, we have what is happening in the brain. The main parts that are affected by the biological system would be the connections to the control state of the emotions. So another way that these two biological systems are connected is that so I am representing the feeling state also as amygdala, and there have been research that in people with PTSD, there has been inflammation of the amygdala. Therefore, there is a line that connects inflammation and the feeling state. Another way that biological model affects the mental model is it strengthens glucocortics so cortisol is very important in strengthening in the connections in the brain in emotional regulation. And so basically in this model, emotional regulation is strengthened by the glucocortics. Yes, okay. So here you can see the Leila van Mentenian-Treur's model. Main changes I made to the model is, so these two states have Hebbian learning because therapy is used as psychotherapy. So the person learns how to control its emotions. In my case, we are using MDMA as a form of therapy to heal DNA differently methylated genes. So because of that, these two states have no longer a combinational function of Hebbian learning, but they have a combinational function of advanced logistic sum, and their speeds have been fixated, so they're no longer adaptive. Another thing about the mental, I think that's it for the states. We can continue. So to the simulations. There are 4 simulations. Scenario 1 is development of post-traumatic stress disorder. Scenario 2 is development of post-traumatic stress disorder, but only of the gene NR3C1. So FKBP5 has been basically it has no effect on the model. And scenario 3, development of post-traumatic stress disorder with only FKBP5. And scenario 4 is the same as development of post-traumatic stress disorder, but there is also introduction of therapy in form of MDMA and symptom reduction. So for the simplicity for this presentation, scenario 1 and 4, I have shown them together. It's because you can see here the development. So right here, this is a healthy person. So from 0.0 to 0.1000, we have a healthy person. After that, a traumatic event is happening. Now the person has developed PTSD, and this is here from 2050 to 2070. Therapy is introduced in form of MDMA. And we can see, and here we can see the difference after the therapy. So for the biological model, so after the event of traumatic event, we can see a decrease in glucocortex, the purple line. We can see an increase in glucocorticoid receptors. We can also see an increase here in inflammation and the immune system. And for the HPA axis, that is the stress response system. So hypothalamus, pituitary gland, and adrenal cortex. The best way to show it would be through the proteins they secrete. So we can see that for CHR, there is, after development of PTSD, there is a decrease in production, and there is a decrease in production of ACTH. So for expression of FK-BP5, we can see that expression of it drops. So that is also a reason that that glucocortic receptors are that is the reason why glucocortic receptors are have higher sensitivity, as there is nothing to suppress it, and expression of NR3C1 is increased, so there's more receptors. Here we can see methylation on these two genes. So we can see an increase in FK-BP5, and we can see a decrease in NR3C1. So for the mental model, we can see that after occurrence of PTSD, there is an increase in all states. That is because there is now a flashback movie occurring every time there is a trigger, while here there is no flashback movie because the traumatic event has not occurred yet. Therefore, there is no flashback movie, so the reaction to the triggers are not that bad. So, for example, we could say here so in a war veteran, fireworks may startle you, but in this case, the person would become very scared of it, and it would remind them of their past experiences. Here we can just see feeling state and the control state, which are two most important that are most affected by my biological model. So we can see that feeling state is decreased so we can see that feeling state is increased after the traumatic event, but the control state is also increased. But we could say that is because now there are more emotions that need to be controlled. So these are the first order states of the mental model. We have also second order states of the mental model. So for the first order state, again, these are so here we can see the weights of the traumatic scene happening inside of the person's brain. This is constant. This cannot be changed. A lot of therapy has been applied, so but it has been noticed that the person will continue experiencing the same video of the traumatic event. The only approach to it is to control your feelings when you remember of it. And these are the states that represent the weights between for the control state. They are increased after the traumatic event has happened, but again, there needs to be more control as there are more feelings. So these are the speeds of the said weights, and they are constant as them. They cannot be changed, as the person will always remember the traumatic event that has happened to them. So let's go back and talk about therapy. So the scenario 4. So after therapy has been applied, methylation of the NR3C1 has returned to normal, and therefore FK-BP5 has also returned to normal. Now, now the theory that this model has been modeled on is that because of the decrease of the methylation on NR3C1 gene, glucocortics, so basically cortisol is low. And because FK-BP5 has been observed as a showcase that how well the levels of the cortisol levels. So basically, we could see how much cortisol does a person have based on expression of FK-BP5. Because there is because there is not enough cortisol because of the methylation on NR3C1, FK-BP5 has been over-methylated, and therefore it's not expressed enough, which further induces the low glucocortics. So let's go back. What happens in the biological model after administration of therapy? All the states go back to normal. We can see it also here. And we can see the expression goes back to normal. Expression of NR3C1 also goes back to normal. And now let's take a look at the mental model. Now, mental model, there is an increase in the control of the emotions, and the feelings have been reduced. That is, after the therapy, the patients have been reported to have their symptoms reduced. That is, they have less of a feeling of the flashback. However, it is not put back to the state of a healthy person, because PTSD is much more than just one demethylated gene, and there are many factors that affect it. So only a reduction in symptoms has been observed. So we can see here the connections are higher. So control state is higher, and connections in the brain are stronger. This is again the video continues, but now the person does not feel as scared of it or as sad about it and such. So for scenario 2, I wanted to test how does the methylation. So basically, I wanted to test each gene individually, and what I noticed is that if FK BP5 has been basically it no longer affects the model. Then NR3C1 does not demethylate, and after exposure to chronic stress, it goes right back to normal. And same thing happens with FKPP5. So for the discussion, I have introduced a computational analysis perspective on epigenetic effects in PTSD. I have introduced a form of therapy in form of MDMA. However, regarding this model, not everything is known on DNA methylation and the biological background of fear processing. For example, the connections in the brain I have mentioned, they have been observed in rodents. They have also been observed in humans, but they weren't really able to pinpoint it as good as in rodents. Both fields we have just recently started to explore, and they will be explored even more as technology improves. MDMA has been tested on a very small sample of individuals, but shows promise. Also, MDMA as a methylator has been shown to methylate in cardiac genes. So it fixes demethylation in case of cardiac genes also, but more tests should be made on MDMA and PTSD. Everything is referenced to a scientific paper. Some claims are just theories for now. For example, the fact that FKBP5 is only overly methylated because of the methylation levels on NR3C1 are reduced. Furthermore, not all PTSD patients have been observed to have low cortisol, and some PTSD patients have been observed to have higher or lower levels of methylations on both of these genes. However, it really depends on age, when the traumatic event happened. It depends on sex. It depends on genetic background. But those are also things that affect will you get post-traumatic stress disorder after it has been after traumatic effect, as not everybody gets it. But in this model, we have focused only on DNA methylation on this specific case. So I think that is everything. And do you have any remarks or questions for me?